Our next finalists are from Miami Lakes, Florida. I'd like to welcome Mark Raymer and Dr. Christine Sapin from Asana Medical. Mark, how you doing?
Great.
Nice to see you.
Nice to see you.
You know, they say Miami has the oranges, but Buffalo has the juice. Remember that.
And the better football team right now.
And a much better football team. Yes, yes, yes. This will prove in 2 weeks, by the way, as we come down here. I'm going to turn it over to you guys. The floor is yours. Here's the cue. And you have 10 minutes. Thanks.
Okay. Good morning. Thanks for your attention this morning. It's an honor to be among the finalists for 43North's inaugural competition. Ulcerative colitis is a debilitating disease of the gastrointestinal tract. Once it strikes and the colon loses its ability to function, it compromises your life in many ways. It really does wreck your quality of life. Right now, the only ways to treat UC are with drugs that can potentially have very serious side effects, or with surgery for those patients who don't respond to the drugs, major surgery to have their colon removed. Neither one of these is obviously an adequate solution. What if I were to tell you there's a better way? What if I were to tell you that someone is developing a medical device that in many cases can minimize the need for these drugs or for this major surgery? Well, that's what Asana Medical is doing. My name is Mark Raymer. I'm the CEO of Asana, and we'd like to spend the next 10 minutes sharing our technology with you. Now, here's the problem. There are about 6 million people who suffer with UC, and there's no treatment method available to them that directly addresses healing of the tissue that's been obliterated from the disease. So that's what our solution is predicated on, a means to directly address healing. We'll elaborate on that in just a moment. The other points that you see on the screen, the strength of our management team, the strength of our IP, and our rapid path to clinical trials are just some of the points that make Asana a compelling investment opportunity. We'll elaborate on those as we go forward. At this point, I'd like to bring up Dr. Christine Sapin to tell you more about the problem and how we're going to solve it.
Thank you, Mark. Good morning, Buffalo. And what a warm welcome. And this is coming from somebody who arrived from Boca Raton, Florida. Ulcerative colitis is a serious autoimmune disease, an inflammatory bowel disease that is growing worldwide. The disease does not discriminate among women, men, or child. Because the prevalence of autoimmune diseases such as MS, diabetes, celiac, and others is fairly prevalent in Western New York, there are centers of excellence to treat such diseases, and we would see a great opportunity for our company to be located here to work on UC. The disease affects the mucosal lining of the colon, destroying its natural ability to function that can lead to severe physical, social, and emotional issues. This slide shows the differences between the normal colon on the left with the smooth mucosal lining and the UC colon on the right with the irregular pattern. The lower pictures of the endoscopic views and pretty much look the same. The quality of life for these patients is greatly diminished because it is a lifelong disease without a cure. UC therapy starts with an anti-inflammatory aspirin-like drug that can rapidly proceed to potent steroids, immunosuppressants, and finally 20 to 30% of all patients have to have their colon removed simply because the drugs do not work. With each drug, the chances of serious side effects also increase. Asana's technology will treat the patient early in the disease cycle to arrest the advancement of ulcerative colitis. Our science is a shift from tradition. We'll be using a device called extracellular matrix, ECM. It is a complex network of proteins found throughout human tissue. FDA has approved many ECM products which have safely treated burns, heart, wounds, nerves, and others. Asana is using a proven technology and applying it in a novel application for UC. Simply stated, ECM stimulates the body to heal itself and grow new tissue where it is applied. It recruits cells, growth factors, and acts as the foundation to transform the diseased tissue into healthy. Asana's ECM product will be delivered non-surgically into the colon using standard GI techniques. What makes me excited about Asana's technology is we have an animal study that showed for the first time ever that ECM promotes rectal mucosal healing, regrowth, and this is exactly what we need to see for UC. 6 animals had surgery to remove the rectal mucosal lining, and ECM was applied to 4. The gross anatomy and histology show that the mucosa in the ECM-treated group on the top is restored to normal, whereas the control animals had no regrowth, as evidenced by the severely scarred tissue. This study simulates the worst case of UC, yet we had a very positive result. This study provides compelling data that ECM works for the growth of colon tissue. In my professional history, I have not seen a more approachable scientific problem that we will be able to convert to a product. And now I'd like to turn it back over to Mark to discuss the business.
Thanks, Christine. So everything we've talked about to this point, the problem and the solution, all just represents potential. But as you all know so well, without the right leadership team, none of this can come to fruition. To that end, I offer Asana's leadership team. Altogether, we offer over a century of experience with a great track record of success, the technical know-how, the business know-how to bring this home. I'm the CEO of the company following a 20-year career as a biomedical engineer. Christine has great depth of experience in the technical aspects of biotech, medtech, et cetera, having been part of major efforts with product development, clinical trials, product launches. Jerry and Rick bring to the table finance and business knowledge. They've helped initiate companies, get them financed, help them grow, and ultimately see them out the door by way of acquisition. More to the point, we've attracted the kinds of advisors that you typically see at larger companies, people that with substantial knowledge and reputations that they put at risk associated with Asana because they know we're doing something special. Case in point, Michael Phelan, a C-level employee at Boston Scientific. Or in the middle of the page, Dr. Stephen Badlak, the deputy director of arguably the world's foremost center for regenerative medicine. At the bottom 6 names comprise our medical advisory board, key opinion leaders on a national, if not international basis, with great depth of knowledge in colorectal surgery, GI, and pathology. So another compelling aspect of Asana is the large market opportunity we're penetrating. Christine mentioned that we're targeting the patients who have failed that first line of drugs, the aspirin-like drugs. All the drugs superior to that are the ones that have the devastating side effects, so there's a hole in the market. That's the unmet market need. We're actively addressing reimbursement, which I'm sure you realize is a critical factor. We've enlisted the support of a reimbursement consultant to help us achieve routine and predictable reimbursement for the hospitals and doctors who use our product. And all told, there's a multi-billion-dollar market opportunity based on the reimbursement rates and the prevalence of UC. Our IP portfolio is strong. We've filed 1 patent with myself as co-inventor. We are in-licensing additional technology to round out our patent portfolio. And we've completed a preliminary landscape review to ensure we're not infringing anybody else's claims. Now, all of this is defined in our roadmap. We've rigorously defined the project plan that's going to spell out the milestones we're going to achieve. They're laid out here. The one I'd like to call your attention to is the 3rd one, demonstration of proof of concept in humans. We're going to achieve this initial proof of safety and efficacy in people within our first 2-year operational window, which will be funded with our initial raise. To that point, we'll be raising $4.25 million, broken down into a $750,000 convertible promissory note, most of which has been spoken for through informal commitments, followed by a Series A preferred stock offering for $3.5 million. Our use of proceeds is also shown with the lion's share of the money going toward product and process development. And again, because it's so important, I'd like to point out that generating this initial human data within 2 years is a real driver for us. That's our first major inflection point. It could even be an exit point for the company. So altogether, we know that Asana represents many of the aspects that investors look for when they're looking for a sound investment opportunity. We have a product with fantastic potential that's been realized by virtue of the initial animal study we've done. We've got a leadership team with great experience and a track record. We've been there and we've done that. We've already launched many products. We've had successful exits. We have a strong IP position. We have a huge untapped market opportunity available to us. We have a clear path to human clinicals and so forth. We know that Asana is onto something special, and we look forward to partnering with 43North on the rest of our journey. Thank you for your attention.
Nicely done. You're lucky I'm not a judge because I understood everything and I would have had some very in-depth questions for you. We're going to start, obviously, David. You get the first question.
The doctor? Really?
Absolutely.
Thank you very much. I want to understand a little bit more about how the initial treatment works before you put in the matrix, and then how matrix is injected or implanted, installed. I presume that you're doing a submucosal resection of a lesion in question using an endoscope. How do you take out the ulcerative colitis lesion, or do you?
No, we're not going to.
Okay.
What you're referring to, just for the sake of the non-scientists in the audience, is a process that is done in the esophagus, for example, with Barrett's esophagus, where there's a precancerous lesion and they pull out that tissue and then somehow fill it in. When you do a 360-degree removal of that precancerous tumor, the tissue scars down, its strictures or narrows, and it can compromise the integrity of the whole organ. We are not going to do this technique in the colon. We're going to infuse this as a hydrogel. It's going to go in as a liquid with a viscosity akin to water, and once it reaches body temperature, it gels. It forms this tenacious, adherent gel over the ulcers, and because it's under visualization, the doctors will be able to see where the most ulcerated parts of the colon are.
Okay, so the lesions are not removed, they're merely injected. How extensive can this treatment be? I mean, ulcerative colitis is frequently a disease that involves multiple spots throughout the length of the colon. Are there limits on the number of lesions that you think you can inject?
We don't think of it in terms of number of lesions per se, but rather we're going to be treating left-sided colitis from the splenic flexure all the way to the rectum. Okay, and we're just talking about the distal part of the anatomy there. This can be readily accessible through endoscopic means, colonoscope, flexible sigmoidoscopy, and again, injected as a liquid. As we learn how to use the technology efficiently and we see proof of concept, we'll expand to be able to do the entire colon through something like a colonoscopic technique.
Okay.
So to follow up on those questions, whatever it is that causes the ulcerative colitis, won't it simply, again, kind of invade the healed tissue? So do you need to do this once a year? Or, I mean, it's great to take it out of the colon and test it in animal studies, but in an operating human being with the conditions that cause the UC, what happens?
It's a great question. That's a $64,000 question that many clinicians have asked us. If a body has colitis and you're going to create healthy tissue, why won't that new tissue become colitic? That's your question, right?
Yeah.
Okay.
Thank you for saying it so nicely.
The way I just want to make sure we were clear. There's something that happens with extracellular matrix, and this has been proven in other applications of ECM around the body, where it undergoes a phenotypical change, a genetic change, if you will, so that it goes from being predisposed to being inflammatory tissue to being predisposed to being natural, reconstructed, healing tissue. Okay? Why this mechanism occurs is not well established, but the fact that it does occur is well established. It's been published. Okay? Now, if that happens, we have a grand slam, because the colon will stay healthy for a very long time. Even if it doesn't happen, though, if we can just induce remission for a little while and give drugs or other therapies a chance to work, that's still a home run for us. Because keeping a patient in remission is way easier than taking a patient from flare to remission.
Right, and just one final thing. So you do this to me, presumably I'm lying on a table and maybe I'm anesthetized. How long does the gel stay inside? I mean, am I in the hospital for 3 days or for 1 hour? What's the...
Excellent question. Yes, it's a tenacious and adherent gel, but as soon as you have a bowel movement, you're probably going to shed it, okay? So it's going to require repeat treatments. How many treatments is yet to be determined. That'll be fleshed out in our animal work and our early human clinical data.
And Mark, just to add to that just a little bit, ulcerative colitis starts as a collection of micro fissures within the colon. So the gel will naturally migrate to the micro fissures and stay there, and because that's its natural inclination. As these fissures get larger and larger, they become ulcerative. And again, that tissue is adherent for this ECM product. So it's an excellent question. Thank you.
strikes me that $4.5 million is a tiny amount of money for what you want to achieve.
A fair point. This is going to keep us operational for just the first 2 years, so it's going to carry us through all of the preclinical work that FDA and other regulatory bodies would require to allow us to do human clinicals. And then the human clinical itself, because it's quite small scale, it's 10 to 20 patients, is not going to be an exorbitant cost. We're really rigorous in defining our project plan and our cost structure. We're confident this is sufficient money. Remember, it's just enough to carry us through this first phase, which will then allow us to increase the value. We'll do a subsequent raise through perhaps the next clinical trial, another raise to get us out the door.
Got it. Okay.
Kim?
He took my question. Oh, that's all right.
I read your mind. I knew it, so I thought I'd just get it in there quickly.
And if I could add to the point about the human clinical studies, there is new regulations from the FDA that does allow us to explore things as a device in the first 10 patients, and we can add on another 10. So when something is as safe as this is, been proven to be safe and effective through other parts of the body, we feel confident we can get into the human studies early, and we obviously would have to follow up with some additional human clinical studies, but this will take us through the early studies.
Kim?
You did talk about your staff and the organization as a whole. So, you know, how many, is it just you two in your group?
No, there's 3 of us who are actually on
us,
staff,
okay.
technically on staff as paid employees of the company.
Over the course of the next 2 years, how do you see that growth in your staff?
Well, we're certainly going to have to ramp up who we hire as we progress along this path, because 3 people alone aren't going to make this happen. But we have a great support team behind us. We have a series of consultants who we work with to support regulatory, QA, testing, manufacturing, for example. That will all be strongly controlled from our from the inner circle within Asana.
Randall?
I mean, to that degree, I saw a lot of your roots are obviously strong in South Florida. You're 2 years away from a human clinical trial, 5 years from a larger rollout. What is your commitment or thoughts on staying in Western New York long term?
Well, we are certainly going to take advantage of every opportunity that 43North makes available to us, the capital infusion, the incubator space, the mentoring, anything that helps Asana grow its value. Buffalo Niagara Medical Campus or Center, I'm not sure which the C stands for, but BNMC offers us a world of opportunity as far as working with GI-based clinicians there, recruiting patients for our clinical trials, and we could tap into the patients who actually suffer UC. And help them in this region, so we could save Buffalonians lost work hours, improve the quality of life. So the point is, we are going to become entrenched in this society up here.
John?
How long have you been working on this?
Asana was incorporated in May of last year.
Great. Thank you.
And just to add to the location, one of our founders is from Buffalo, and our other founder is, as we speak almost, looking for an apartment. So if anybody has rental spaces, we're looking.
No, they need to buy it.
Oh, good idea.
David?
Yeah, given that one of the major challenges in managing ulcerative colitis is the transition from inflammation to cancer. Significant incidents, as I'm sure you're well aware. And what are your thoughts about the longer-term trial aspects of this? Because I think, assuming you treat 10 or 20 patients, isn't the next question going to be, what about mitigation of the risk of malignancy? Is there any effect? And then what kind of trial and development sequence do you see, assuming you reach that first or second or third milestone, whatever it is, in your initial group of patients?
Christine, you want to take that?
Yeah, excellent question again. Thank you so much. The plan is to have really essentially lifelong follow of all the patients that we treat. You know, being in this business for a lot of years, that lifelong data will give us answers, and obviously the more patients that we recruit, the longer we'll have data for, and the more data. You know, relative to the cancer, that is on our radar screen without a doubt. It's a very serious problem with these patients, and we want to see if putting patients into very long-term remission, if we can reduce that cancer risk. So that's an interesting combination there.
Can you just give us the numbers, the current numbers of after 20 years or something? What's the cancer rate?
Oh, yes. It increases by every 10 years, it goes up another, I think it's 5 to 10%. So, it goes up in decades. And remember, this is a lifelong disease. It can start in children. And so for their whole life, every decade, it goes up by a significant percentage. So by the time if somebody gets it 15, by the time they're 65, the risks are very high.
If they make it.
So you do anticipate a major follow-on trial after your initial demonstration, I presume? Is that a fair assumption?
Yes, we will be doing a pivotal study for efficacy, safety.
Great. Thank you both very much.
Thank
Christine.
you so much. Pleasure. Thank you.
Mark, thanks a lot. Well done.
Thank
well
you.
done. Thank you both.